My name is Jim, and I work as the Global Application Specialist for Cellink. Today I will talk about bioprinting skin tissue models. I will first give you just a brief background to Cellink, who we are and what we do, then move on to more practical how you can print a skin tissue model. So, Cellink as a company is very young. We were founded in 2016, so we just turned 4 years old, but we already have customers in 60 countries, 6 continents. We have a lot of offices now. Our headquarters are in Sweden, in Gothenburg, and that's where the company was founded. We have 2 locations in the US, several locations now in Europe, also one in Kyoto, Japan. So we are all over the world. And even though we are very young, we are more than 200 people already. We have our products in more than 800 labs. So the expansion has been very, very rapid. And you might wonder why would we bioprint anything in the first place? So in the very, very long run, the hope is that we can bioprint organs, a heart or liver and so on. But that is in the very long run. In the closer time span, already now, when we can bioprint something, we could potentially reduce the use of animals. For example, if we take skin, for example, the cosmetic industries are not allowed to test their products on animals in many countries. That's where bioprinting of a human skin tissue model comes in very, very handy. And basically what you need to be able to do this is that you need to have your cells. You need to mix those cells with a bioink or a biomaterial. That can be done with our cell mixer, where you just put the 2 materials in 2 syringes and push down so that the cells are homogeneously mixed into this cartridge. And you put the cartridge in the machine and you can start printing your skin model. That's the very, very basic principle. In addition to the bioprinters we have a few other machines as well, like a single cell dispenser, where you can dispense individual cells into 96 well plates or 384 well plates. It's very good for cell line development, antibody development, single cell genomics and proteomics. We also have a low volume dispensing machine that can dispense both cells and chemicals, drugs, substances, and so on. And also, when you're working with cells, you would like to see what the cells are doing in the incubator. So for that purpose, we have developed the live cell imaging system, where you can put your cells and the machine inside the incubator and follow it live, exactly how they develop. So basically, we're trying to have a full set of equipment that you might need in your cell culture lab. Today, however, we will focus on bioprinting and specifically bioprinting skin. So I already mentioned that you need a bioink or a biomaterial. So this is basically to support the cells during the printing process and after the print is done. We have today more than 50 different bioinks, and the reason for that is that we have so many users that are using many different cell types, and a heart cell needs a very different material compared to a liver cell or a skin cell, for example. So we need to have all these different materials to have something to offer for all our customers. Then you also need the 3D model itself. That's basically what will instruct the machine to print your model. So this can be a 3D model that you make in a CAD software It can be a G code if you're a little bit more advanced user. But that's basically to tell the machine what you want to print. And then, of course, you need the machine itself. What you see here is our flagship bioprinter, the BioX. It's got 3 print heads, so you can print up to 3 different cell types or materials. If that's not enough, we are developing and soon releasing a 6 print head BioX. For example, when you're printing a skin model, if you want to include more cell types, you might want to use fibroblasts, keratinocytes, melanocytes, endothelial cells. You might want to use one of our other tool heads for the camera for taking pictures of your prints or for photo cross-linking. So then the 6 print head printer will come in very, very handy. We have a couple of other bioprinters as well that uses a light-based technology. These are very good for printing structures with a finer resolution and good for vasculature printing. But today it will be extrusion-based bioprinting, skin tissue models. And I briefly mentioned why this could be a good idea. Reducing the use of animals are good not only for the animals, but since we also can use human cells, so then we can print a model that is more similar to the human body, meaning that we can get more relevant data from our printed model compared to the animal model, because an animal is not a human being. So we should be able to get more relevant data from our printed model. And this is one of our models that we have been printing in-house. And the idea here is to mimic the basic layers of the skin. So we have printed first a couple of layers of a low concentration of fibroblasts, and then a high concentration of fibroblasts on top. And then the top layer is a high concentration of keratinocytes. And this is basically the basic parts of the different skin layers. So a fairly simple model, but you can, of course, design it differently. You can have different layers, you can have different cell types. This could be a good model for compound testing. You print your model, culture it, add your compound, and analyze what the cells are doing. So fairly simple. But there are a lot of advantages of these bioprinted skin models, because you can print them whenever you want. As long as you have your cells ready, you can print them at any time. You can adjust them in size. If you want to print a big skin patch or smaller ones, you can decide that yourself. You can also decide how many you want to print. And this is very, very important, the donor-specific part. You can actually use cells from specific patients, because a skin biopsy is fairly simple to do. If you want access to a person's liver cells or heart cells, that's not very easy when the person is alive. So a skin sample from a specific donor is a simple procedure. You also have the advantage of using a machine, because then you will print the same over and over again. So instead of doing it manually, where you have a lot of variation, you can have the machine do this for you. So there's a lot of advantages of bioprinting a model like this. So this is basically to summarize the idea behind it. You have your 3D model, you mix your cells with the ink. We have used and developed 2 separate bioinks specifically designed for skin. They're called Cellink skin and Gelexa skin. Both of them contain fibrinogen, that when you cross-link them with thrombin, is converted to fibrin which is an important part of the skin. The Cellink skin is based on cellulose and alginate. Gelexa skin is based on methylated gelatin with added xanthan gum and alginate. The advantage of Gelexa skin is that you can use both UV light and ionic cross-linking, so you can play around a little bit more with the stiffness of the construct. Then it's basically up to you what kind of cells you want to use. If you want to use fibroblasts, keratinocytes, melanocytes, endothelial cells, it's completely open. In this very example, we printed these models into a 24-well plate. We cultured them fully submerged in media for 1 day. Then they were flipped and put into these inserts, simply to lift the cells up to the air-liquid interface. That's simply to better mimic how human skin develops, because the human skin is, the top here is exposed to air. So that's what we're trying to mimic by culturing them in these inserts. And this is just a video how it can look. This is actually another model where we have used only 2 print heads. So we print a few layers in the bottom and then like an edge around. So in here we could potentially seed cells manually. If you want a very high concentration of keratinocytes alone without bioink, you can add them manually. But this is basically how simple it is. Once you have your ink, your cells mixed, you press play on the machine and it will create the models for you. It's fairly simple. This is how it can look when you have printed a plate. This is just a summary of what I mentioned, that we culture them submerged for 1 day, lift them up to the air-liquid interface, and then of course you need to analyze your construct to see that the cells survived in the first place and that they are responding to treatments and that they look the way they should morphology-wise and so on. So I will now show you some examples of analysis that we have done and that you can do. But these are only examples, so there's plenty of other methods that you can use. But to give you an idea, so in the first example here, we used Cellink skin, and the 2 pictures to the left is just a simple live dead staining. It's a commercially available staining kit. You add this to the construct, and then the live cells will turn green, the dead cells will turn red. There are some dead cells. It's not that easy to see here, but there are some dead cells, but most of the cells, they do survive, which is what they have to do, otherwise we have nothing to work with. And in this very example, we also had access to, we could take some multi-photon images, basically to see the morphology of the fibroblasts here. And what we want to see is that they really stretch out and that the fibers like intertwine to form a thick network of cells, because the skin needs to be very, very tough, so you need a dense network of fibroblasts. And also the keratinocytes, we see that they proliferate and they start to self-assemble into, tissue-like structure, because the keratinocyte layer has to be quite tough as well. So it's good indications that they proliferate and self-assemble into tissue-like structures. In the second example, we have again live dead stainings in the top of cells printed in Gelexa skin. So these are different from the multi-photon images, but we have a good survival of the cells. And in this case, we also cultured them for 28 days. We added a compound that is designed to organize the secreted collagen into networks. So in the non-treated version, we can see that the collagen is dispersed all over the construct. In the treated one, we can clearly see that the collagen have formed more clear networks, which means that the cells are responding to the treatment the way they should. So very, very nice indications. And if you don't have access to, so you can take multiphoton images, if you still want to see the morphology of the fibroblasts, you can do a more simple actin staining to see the network of fibroblasts forming. That's the same staining as on the previous picture. Of course, you can also do a simple H&E staining to get a more overview of the morphology of your printed construct. We have also, I mean, there's so many different things you can look at. We wanted to see also the expression of filaggrin and keratin 10. And when it comes to keratin 10, that is basically a marker, proliferation marker for keratinocytes. And the first example to the left, we have cultured the cells fully submerged for the period of culture. In the middle, we have cultured the cells in the air-liquid interface. And what we could see was that in the submerged culture, we have the keratin expression evenly dispersed over the construct. And in the air-liquid interface, we see more keratin 10 expression closer to the dermis layer. And that's important because in human skin, the keratinocytes basically, they mainly proliferate close to the dermis layer, and then they migrate towards the top of the skin and die. Because the very outermost layer of the skin is dead skin cells. So we saw in this example that most of the keratin 10 are found closer to the dermis. So they seem to behave more like human skin in that example. And again, of course, you can also do morphology staining in these cases as well. To make it easier for customers that want to print skin, we have developed these kits. So the kits will include one of the two skin inks that I mentioned, and also 3 antibodies, so that you can do 3 stainings if you would like. So we have antibodies for collagen 1, elastin, and keratin 10, 3 important components in the skin. And you can, of course, combine these with other analysis as well, but we think these are 3 of the key components that you might want to look at. And we could see when we stain our printed constructs that this is collagen, and we do have collagen. This is just the negative control, so there are some background staining. This is actual human skin, so we can see that morphology-wise, we're not quite there yet. But for the antibody, it works and it stains the collagen. We see the same basically for keratin 10. It's possible to stain our printed construct. We can see the keratin 10. Same for elastin. So these pictures is just, this was experiments to verify that the antibodies worked on a 3D printed construct. And that we could see. So that's a good step. So these were all done on a fairly simple 3D printed model. If you want to print something more complex, you can add more cell types. You might also want to add vasculature, because if you want to print bigger structures, you will have problems with oxygen and nutrient delivery to the center of the constructs. So to avoid that, you could print a channel through the construct and perfuse with either culture media or culture media plus cells. And we have developed a kit to help you with this as well. So this is called the VAS kit. It's basically designed so that you, in a very simple way, can print a channel through your construct. So to add a little complexity to your printed construct. This nice little video that you will see shows you exactly how it can be done. This is just an example. You can do it in many other shapes and forms and so on. But the idea is it's very simple. You will see one of our old printers here. But the idea is the same which printer you would use. You would basically print a tube with a sacrificial material. In this case, it's Pluronics. Pluronics is very good because it becomes a liquid when you cool it down. So you can print this tube, you can cover it either with a bioprinter or manually, as we did here. And then when you cross-link your real bioink, you can lower the temperature and basically aspirate the Pluronics away. So you create a hollow tube through the construct. So here we pipette Gelma on, but you can easily use this in a bioprinter as well. So here we aspirate the Pluronics away. You see the nice channel. And then you simply connect the device to a perfusion pump, and you can start perfusing it with media, for example. And I mean, to perfuse it with media is good for, then you ensure good oxygen and nutrient delivery to the whole construct. You can also add cells, for example, endothelial cells, and try to have them attached to the inside of the tube, and then start forming a vessel wall so that you would have something more similar to an actual blood vessel. There are many other ways of doing this, but this is one quite simple way of doing it. So now you know how to make a simple and a slightly more complex skin model. And by that, I would like to welcome you all to the world of bioprinting. Thank you.